Our names are Sophia and Venetia, and today we are going to be discovering about meiosis in plant cells. Meiosis is a process that sex cells, or gametes, undergo. Through the process of meiosis, a cell ends up with half the number of chromosomes of the original cell. Meiosis 1 begins with a diploid parent cell, which means the cell has 2 copies of each chromosome. Before the process of meiosis, interphase occurs. The DNA in a cell's nucleus duplicates and the cell grows. After interphase, prophase 1 begins. In this stage, the strands of DNA, called chromatin, begin to coil and form 2 pairs of homologous chromosomes. Towards the end of this stage, the nuclear envelope starts to disappear. The spindles attach to the centromere of each chromosome. Some of the chromosomes in homologous pairs cross over during this process. Crossing over is the process that happens when homologous chromosomes pair up and exchange different segments of genetic material. This happens in order to contribute to genetic diversity, which then makes for genetically healthier offspring.
Next, in metaphase 1, the nuclear envelope is completely gone and the homologous chromosomes start to line up in the middle of the cell. Unlike in mitosis where the sister chromatids get pulled apart, in meiosis the sister chromatids stay together and the homologous pair gets pulled. In metaphase of mitosis, the chromosomes line up individually, while in metaphase 1 of meiosis, the chromosomes line up in pairs to form a tetrad. These homologous pairs get pulled to either side of the cell by the spindle fibers. In anaphase 1, the chromosomes are fully pulled apart to opposite sides of the cell, with the spindle fibers still attached. And then in telophase 1, there is a complete haploid set of chromosomes at each pole of the cell. A cell plate in each cell starts to form and 2 nuclear envelopes begin to reform. By the end of the stage, 2 daughter cells have formed. Cytokinesis occurs and the cytoplasm of the 2 cells is separated. Moving into meiosis 2, the cells sometimes get a rest period during interphase, but usually move into prophase 2 fairly quickly. In prophase 2, the nuclear envelopes of both cells begin to dissolve. The spindle fibers then begin to form and grow. In metaphase 2, the spindle fibers attach to the centromere of the chromosomes, and the dense chromosomes line up in the middle just like in mitosis. During anaphase 2, these sister chromatids get pulled apart to opposite poles of the cell. Finally, in telophase 2, a cell plate forms and the cells are pulled apart. Cytokinesis occurs and the cytoplasm of each cell is split. 4 daughter cells are formed in total. At the end, each daughter cell contains 2 sister chromatids. After they separate, chromatids are now called chromosomes. The nuclear envelope reforms and the DNA uncoils into chromatin.
Thanks for watching. We hope you learn a lot about meiosis.